discuss about Human Genome Project. Human Genome Project, commonly abbreviated as HGP. 1950s and 60s saw significant developments in the field of molecular biology. The structure of DNA was discovered, genetic code was discovered, gene regulation mechanism was discovered. So slowly scientists started understanding the molecular basis of inheritance. With more and more information coming on DNA, more and more curiosity developed on the role of genes in the pattern of inheritance. What is the exact number of genes in the human DNA? What characteristics do they express, and so on. So, in the year 1990, the National Institute of Health of US government, along with cooperation from UK and many other countries, started a huge project, a collaborative project to understand the complexity of human genome. The word genome is used to describe the complete set of genetic information that is present in the haploid set of chromosomes. So, the total genetic information present in the 23 chromosomes in human cell is known as human genome. So, this project was named as Human Genome Project. Initially, it was thought that the project would take about 15 to 20 years to know the details about human genome, but a little earlier, by 2003 itself, within 13 years, it was possible for scientists to understand various aspects of human genome. The main objectives of this Human Genome Project are as follows. The human DNA, according to an estimate, contains nearly 3.2 billion base pairs. According to an estimate, the human DNA in every cell has about 3.2 billion base pairs. So, one of the main objectives of this Human Genome Project is to find out the sequence of this large number of base pairs. So what is the exact sequence of these 3.2 billion base pairs? How many adenine, how many thymine, how many guanine, how many cytosine, nitrogen bases are present? What is the exact sequence? This was one of the main aim. Number 2, to find out the approximate number of genes, in the human DNA. If you take the DNA from a cell, unwind it, it can reach a length of about 2 meters, containing 3.2 billion base pairs. But all these base pairs or the entire length of DNA does not function in the form of genes, only specific regions. So another important aim was to find out the approximate number of genes in the human DNA. Then another important aim was to find out the characteristics expressed by these genes. Once the approximate number of genes is known, what characters do they express? So how many hereditary characteristics are present in human beings? Another important aim was to store this information in the form of databases. To store the information in the form of databases, another important aim was to provide this database to laboratories and industries that are involved in research and production related to human health and one more important aim of Human Genome Project was to address the ethical, legal and social issues. Ethical, legal and social issues, together called LC, that may arise from the project, mainly because there could be various kinds of objections. What if the data could be misused? legal problems associated, social problems associated, because we have so many social categories of human beings. What if all those genetic information is misused? So these were some of the prime considerations for the Human Genome Project.